Well, the initial reaction was surprise and that I should be chosen for such an honor, and obviously I'm extremely pleased and somewhat, I would say, still getting used to the idea. It's a wonderful thing to be awarded, and I'm extremely grateful to everybody who was involved in that. Well, we know what the genome is. It's the sequence of bases in the DNA that contains the information for making an organism, including a human. The questions that we don't know the answer to fully are how that information is released, how it is controlled. And so on top of the information in the genome, in the DNA, are layers of information that allow the DNA sequences to be read appropriately. And one of those layers is DNA methylation, which is basically the addition of a chemical group, a small chemical group, to the DNA after the DNA is synthesized, and this is in a pattern. And our first contribution really was to be able to map where DNA methylation was, because one can't understand something like DNA methylation unless you know where it is. So my group initially developed a way of mapping, and that's how we got into this area. So we know that DNA methylation is organized in a pattern, and that pattern is slightly different in different cells in the body. But in cancer cells, which are essentially a parasite derived from your own body cells growing in the body, we know that DNA methylation patterns are really messed up. And in particular, there's a lot of interest in the fact that certain genes that depend on the absence of methylation for them to work become methylated, and as a consequence of that, they are shut down. So there's although the relationship between DNA methylation and cancer is not fully understood, it seems pretty clear that some genes are switched off that should not be switched off, and this facilitates the development of cancer. Interest in Rett syndrome arose really by accident. We were studying a protein that reads DNA methylation, and it turns out that mutations in the gene for that protein cause Rett syndrome. Almost all Rett syndrome is caused by that mutation. So the first thing we did was try to make a model of an animal model of that disorder. And that recapitulates human Rett syndrome rather well. And the next question, since Rett syndrome is not accompanied by neuronal death, like neurodegenerative disorders, but the neurons are still alive, even if somewhat simpler, we asked, if you put MECP2 back, do they become fully functional neurons again? And your question is whether or not the result was surprising, and the answer is it was very surprising, because everybody, including us, expected that it would make no difference, that it was too late. And so when we didn't have funding for this experiment, we didn't think we could get any funding, so we did it on the side. But the result was spectacular. It turned out if you put back the protein, then the mice recover completely. And the result was very clear. And what it means is that Rett syndrome is potentially also in humans reversible and therefore curable. And it was a surprise to everybody involved. And it does make one ask the question for other neurological disorders, which we tend to view as irrevocable and fixed and untreatable. Could it be that they too would be susceptible to therapy. So I think it was one of the results, not the only result perhaps, that contributed to a new idea that maybe mental illness of various sorts may not be as untreatable as we originally thought. I am a basic researcher in that I'm interested in how DNA methylation works. I'm interested in how genes are regulated, and these are fundamental questions. But human disease quite often involves mistakes in these basic processes. Actually, I was quite happy working without any relevance for human disease, but once I realized that there was a connection, then that actually was quite a thrill, and it injected an urgency into our research that I've certainly found very stimulating, and I would never want to lose that. So now I don't see myself only as a basic researcher, but as someone who's interested in biology and the way in which it can impact disease. And Rett syndrome has really provided us with that, and I would never want to lose that.